This video covers the IGCSE Biology Objective 2.1.2 on the 2025 CAIE syllabus to describe the structure of a bacteria cell. So first of all, let's talk about the classification of bacteria. So there are four general components that would determine the classification of bacteria. That is the cell wall composition, what the cell wall is made out of, the gas requirements for the bacteria, the nutritional patterns of the bacteria, and the shape. So for this level of biology, we're only going to focus on the shape, and there are generally three shapes that bacteria come in. The first one is called a spirochete, or a spiral-shaped. There is a bacillus or rod-shaped bacteria, and then what is called coccus, or round-shaped. And so maybe the coccus name sounds familiar to some infections that you may have heard of. There are in fact two categories of coccus bacteria. The first one is called a streptococcus that causes strep throat and pneumonia. And they are linked together in a chain. So it's a round shape, and the bacteria, which are round shaped, are linked together in a chain as pictured here. The other one, called staphylococcus, which causes things like skin infections and sepsis, which is internal infections, are also round shaped, or coccal bacteria, but they are clumped together in a group rather than in a chain. So these are the ways that bacteria is classified that you need to know by their shape: spiral-shaped, rod-shaped, and round-shaped bacteria. Strepto in front of the coccus means that they're in a chain linked together, and staph in front of the coccus means that they're grouped together in a clump. And so these are just the shapes of the bacteria themselves, which you probably would not see under a microscope. This is under a compound light microscope. This is a bit more magnification that would require a different type of microscope. So you would see this in diagrams of the bacteria. All right, so let's take a look at the bacteria cell itself and see what the parts are. We've got a cell wall that is generally, now I did say that the material of the cell wall is one of the ways that bacteria can be classified, but for you at this level, you just need to know that it is a cell wall made of a protein called peptidoglycan. It is not cellulose, so on an exam question, do not write a cellulose cell wall. You can just write a cell wall if you are labeling it and cannot remember peptidoglycan, which is a protein material. There is a cell membrane. There is something called a plasmid DNA, which we will talk about a little bit later in the syllabus. There is something called circular DNA, which is just basically the general DNA of the bacteria cell. There are ribosomes and cytoplasm. So these are the parts of the bacteria cell that you should know and be able to label if you were given a diagram to label. It's very important to distinguish between the normal DNA that typically would be found inside a nucleus. However, it is not in a bacteria cell, it's just part of the cytoplasm. And then this small, teeny, ring-like structure called plasmid DNA. Now, bacteria are classified into the kingdom prokaryote. Pro meaning before, and karyote referring to nucleus. So the word prokaryote means just literally before nucleus. And what this means is that the DNA is not separated from the rest of the cell itself. So if you recall in the plant and animal cell that we looked at in the previous video, there was a structure, a nucleus, where DNA would be contained inside. And in bacteria cells, they do not have this separation. It's just part of the whole entire organelle structure in the cytoplasm of the bacteria cell.